Hello, I'm Renata Fu. My study is titled Super3Path Identifying Commonly Enriched Biological Pathways Using 3 Pathway Databases. Genome-wide analysis studies, or GWAS studies, have drastically increased in popularity in recent years. As shown in the graph, less than 400 GWAS studies were published in 2007, while over 3,000 were published 10 years later. The abundance of GWAS studies means that pathway analysis is more popular and important than ever, since it is used to contextualize mutations and differential regulation, thus enabling a more accurate view of diseases and cell phenotypes. Gene set enrichment analysis, or GSCA, is a popular pathway analysis method, as it takes small changes in gene expression into account and have a very low bias rate compared to other methods. Currently, there is not a standardized pathway naming system available since different databases classify pathways differently. Therefore, pathways that are essentially biological equivalents may have different names across databases. This means most pathway analyses are conducted with a single database, and database bias is common in pathway analysis tests. As shown in the table here, primary pathway databases are much more popular than integrated databases, and KEGG is by far the most widely used database. Researchers greatly fail to employ multiple databases in their studies currently, making pathway analysis not as reliable as it can be. To address this issue, the present study aims to develop a publicly accessible and highly automated tool to perform GSCA using 3 databases and to compare GSCA results and return a list of pathways presented by multiple databases. We developed Super3Path, a publicly available GitHub repository accessible through this link. Super3Path streamlines the process of using 3 databases for GSCA and shows pathways presented by multiple databases. It is user-friendly and runs on the command line. Super3Path has 3 modules. The first is an R script that downloads or updates packages required for subsequent steps. The second module is an R script that performs GSCA with the databases KEGG, Reactome, and Wiki pathways. It takes an input.txt gene expression profile with columns for standard gene name and a rankable value, such as log2 fold change. It will output GSCA results to be used by the next step, and it can plot GSCA graphs based on user choice. The third module is a Python script that identifies biological equivalent pathways between GSCA results from each database, using ComPath mapping catalog. By default, it uses GSCA result files from Module 2, but user input GSCA results can be used by specifying command line arguments. The script identifies equivalent and hierarchical or product relationships for pathways of different databases. An example final result is shown here. As shown, a Heron's junction from KEGG is essentially equivalent to a Heron's junction interactions from Reactome, although they have different names. We tested Super3Path with 3 microarray profiles that compare gene expression in ovarian cancer stem cells versus ovarian cancer bulk cells. These datasets, namely GSE33874, GSE28799, and GSE82304, were downloaded from the Gene Expression Omnibus, or GEO. Super3Path returned 5 pathways common to all 3 datasets and presented by all databases: DNA replication, glutathione metabolism, hedgehog signaling pathway, MAPK signaling pathway, and mTOR signaling pathway. All these pathways have been previously correlated to cancer development, ovarian cancer, and/or cancer stem cell, validating Super3Path as a reliable tool. In this study, we have developed a tool, Super3Path, which has successfully identified significant pathways related to the case study phenotype. Super3Path can encourage use of multiple databases as it is fast and highly automated, and using Super3Path will allow partial elimination of pathway database bias. However, currently Super3Path is limited to the 3 databases used by ComPath mapping catalog. We are currently expanding Super3Path's function to analyze more popular databases than the 3 included in ComPath mapping catalog, as well as to create a shiny web app for easier accessibility for the users. Thank you for listening to this presentation.